Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture I'm going to talk about helper T cell in detail. So this is actually the video lecture which is regarded to T cell biology, and if you guys remember, we talked about the T cell biology in detail, and in T cell biology we talked about the overview of the T cell biology, and after that we talked about the T cell receptor, and moreover we also talked about the different types of the T cell. Now this is actually the fourth video lecture or third video lecture which is regarded to T cell biology, and in this video lecture we are going to talk about helper T cell in detail. So let's start. Okay friend, so now let me write that helper T cell. The second name of the helper T cell, or we also call that CD4 T cell or CD4 T lymphocyte, CD4 positive T cell or T lymphocyte. Okay, now the abbreviated form of the T helper T cell is T helper cell. Okay, some people write it HTC. It is wrong. The best abbreviation is T helper cell, TH. Okay, so it is actually the abbreviated form of the helper T cell, and this form is wrong because a lot of my friends and my subscribers says that HTC is a helper T cell, but actually it is not the best abbreviated form of the helper T cell. So now let's talk about the helper T cell. Helper T cell is the type of amino cells, okay, which help to eliminate different kinds of infection inside the body, or we can say helper T cell, as the name suggests, it help to activate different kinds of amino system. You know, there are different kinds of immune system inside our body, like antibody mediated amino system, we can say that cell mediated amino system, we can say that humoral mediated amino system, we can say that antibody mediated system, a complement system. All these are actually the amino system. Now all these system which will be activated when there is any kind of infection occur. Okay, so it is done by the helper T cell. So helper T cell is actually deciding that what kind of amino system will be activated. Okay, let's suppose if I explain it with the help of diagram. So let's suppose if this is the cell, normal cell of the body, as you can see, innate cell or any part of the body cell. So let's suppose in this case, if this is the macrophages or dendritic cell or innate cell of the immune cell or tissue cell or epithelial cell, whatever you can say, let's suppose this cell got infected with bacteria, virus or fungi or any parasitic infection. If this cell got infected, so then what happened? This infected cell, this is the normal cell, and once it got infected, so then it will secrete different kinds of interleukin, as you can see. Interleukins, or we can say cytokines, they will secrete different kinds of the cytokines. Now what are the job of the cytokines? They can attract cells. In this case they will attract helper T cells. Okay, so here in this case they will present or they will mainly present the antigen fragment on their receptor, as you can see. So here at that position they will present the portion or the fragment of the antigen. After that, once the the cytokines are released, so these cytokines will attract a helper T cell. So let's suppose this is the helper T cell. So helper T cell will recognize the antigen portion with the help of their receptor, as you can see. Once they recognize the antigen portion, so then this helper T cell will decide that which type of immune system will be activated. They actually secrete different kinds of cytokines, due to which sometime macrophages will activate it. We will talk to it later that what kind of cytokine will be produced and what are their function. They will sometimes produce those kind of cytokines which can activate the macrophages. They will sometimes produce those kind of cytokines which can activate the B cell, and B cell will then produce the antibodies. They will activate those kind of cytokines which can, activate the epithelial cells to produce antimicrobial peptides. They will secrete those kinds of the cytokines which can activate the complement system, depending upon the type of the infection. Okay. Sometime what happen, this helper T cell will convert it into two cells which we call that effector cells and memory cells. So let me draw here. So let's suppose this is another cell. So this is the memory cell. Okay. And they also convert it into another cell which we call that effector cell. So let's suppose this is the effector cell. This is actually the T helper cell. Okay. Memory T cell, just like memory B cell, we already talked about memory B cell. So memory T cell is also important. Why? Because they can store the information of the, this specific antigen or this specific pathogen. They will store the information of the pathogen, that what kind of damages is caused by that pathogen. Once they store the information, so then it will be converted to memory T cell. The second time, if the same infection come, so then this memory T cell will directly kill that without any kind of thinking. They will directly kill. But this is the second chance. A second chance they will directly kill. But at the first time, it is very slow process. But at the second time, it's quick process. Helper T cell can perform their function very fastly of the previous pathogen. If the pathogen is the previous, so the process will be the fast. If the pathogen is the new, so the process will be very low. So means that they will first form the memory T cell, and after that the process are under the control. This is whole story about the memory T cell. I already talked about that. But the next cell is actually the effector T cell. This effector T cells, as the name suggests, they will secrete different kinds of the cytokines that will affect on the cells. Amino cells, means that they will activate macrophages sometime, or they will activate, this is actually the immature, I forget the very basic things before that. This is actually the immature helper T cell or naive helper T cell, we can say like that. When the naive helper T cell recognize this antigen fragment, okay, once they recognize this fragment of the antigen, so after that it will be converted to helper T cell, and then the rest of the process will occur. Now this effector T cell, as you can see, this effector T cell will secrete different kinds of cytokines, due to which different kinds of immune system will be activated. So this is a little bit story about the helper T cell. Now there are different types of the helper T cell. Let's talk about that. Like we have T helper cell 1, T helper cell 2, T helper cell 17, follicular T helper cell, and we have T regulatory cell. These all are the type of the helper T cell. In the next video we will explain T regulatory cell, okay, because it is very long topic, so that's why here I am not going to explain it. The rest of the 4 helper T cell we will explain it in this video, okay, because it's not too much complicated. But this one topic is a little bit complicated, that's why I will explain it in the next video lecture. Now, why this is the question arises in our mind, that why the body of a human body or organism body need all these helper T cells? Because there is different kinds of infection. There is some kind of infection due to which helper T cell 1 is needed. There is some kind of infection through which helper T cell 2 needed. There is some kind of infection through which helper T cell 17 needed. Similarly, follicular T helper cell is needed when there is another kind of HIV or lupus kinds of infection or diseases are occur. Okay, so these kinds of different kinds of disease, different kinds of infection need each, or we can say any one of the helper T cell. That's why all the helper T cell are very important. Okay, now let's talk about helper T cell in much more detail. First of all, let me explain the helper T cell 1. T helper cell 1. Okay. You know, this type 1 helper T cell will be activated against intracellular infection. So it will be activated against intracellular infection means if the cell got infected, okay, either viral or bacterial. Viral or bacterial. So it means that helper T cell 1 will be activated at that time when there is a bacterial or viral infection or any intracellular infection occurs. So then helper T cell 1 will get activated. Normally what happen in, sorry, not normally, in disease condition what happen, or infection condition what happen, this is a, let's suppose the normal cell of the body, as you can see. If this normal cell of the body get infected with the bacteria or viruses, so then what happen, this infected cell will, or disturbed cell will secrete some kinds of interleukin, which we call that interleukin 12. So let me write, so they will secrete interleukin 12. This infected cell, this is actually infected cell, okay, so they will secrete interleukin 12. Interleukin 12 will activate the naive helper T cell. So naive helper T cell will come, and so this is, let's suppose, the receptor, as you can see, and from that receptor this cell will express the portion of the antigen. So let's suppose this is the, now this is the naive helper T cell. Naive helper T cell once recognize this antigen fragment, so then this interleukin 2 will, again fall down on this naive helper T cell. After that, the naive helper T cell will be converted into helper T cell 1 or mature T helper cell 1. So now it is converted, as you can see. Once it is converted, so then this helper T cell 1 secretes cytokines. So it will secrete interferon gamma and interleukin 2. Okay, this interleukin 2 will activate the other T cell like cytotoxic T lymphocyte or T regulatory cell, whatever you can say. This interleukin will activate this kind of cells. If let's suppose if this cell is got infected with the viruses, so then they will secrete interleukin 2, due to which cytotoxic T lymphocyte will get activated and they will kill the cell. How? I will explain it later. Then this, if there is a lot of amino cells are present, due to which hyper regulatory amino cells are get activated, and autoimmune disease, there is a chance of autoimmune diseases, so then T regulatory cell will activate it and they will control that process. So this is the job of interleukin 2. Then interferon gamma, this interferon gamma will activate the macrophages, and all of you better know macrophages are what? These are the phagocytic cell. And if this is the bacterial infection or viral infection, so then this macrophages will engulf these kinds of microorganism, means bacterial cell, they will engulf it and degrade it and kill each kind of the bacteria by phagocytosis process. Okay, sometime what happen, if this macrophages failed to engulf the phagocytic or failed to engulf this bacterial cell, so then what happen, this interferon gamma will also activate the B cell. Okay, so let's suppose this is what, this is the B cell. Now this B cell will be converted into plasma cell. Now once it is converted into plasma cell, then plasma cell will secrete different kinds of antibody, as you can see. Okay, now these antibody are against each infection, means bacterial infection. So if this macrophage is filled to engulf the bacteria, after that this interferon gamma will activate the B cell convert into plasma cell will secrete antibody. After that what happen, this antibody will coat the bacterial cell. Let's suppose this is the pathogenic or bacterial cell, let's suppose. So the antibody will be coated around each bacterial cell, as you can see. After that what happen, this bacterial cell is looking tasty for the macrophage, then macrophages will easily engulf it. This is the second killing of the bacterial process, how it will be killed by the macrophages. So it means that each antibody will enhance the phagocytosis process through opsonization. This is actually opsonization process. So due to this way, the macrophages will engulf it. So means the phagocytosis process will be done, and it will be enhanced by this opsonization process. Okay, so in this way, the infection will be controlled, or the bacterial infection or viral infection will be controlled. So this is a little bit information or brief introduction about the helper T1, that how they perform their function inside the immuno system and why it is very important and what kind of interleukin they are producing, each and everything. So now let's talk about the helper T2. T helper cell 2. At what time helper T cell 2 will be activated? This helper T cell 2 will be activated at that time when there is parasitic infection. Okay, so it will be activated against parasitic infection, or we can say against helminths. Thus, okay, means it's a kind of worm. If there is worm like infection occur inside the body, so then helper T cell 2 will get activated. Let's talk about that in detail. What happened? Let's suppose if this is again the normal cell of the body, and if it got infected, innate means innate cell of the body, or normal cell of the body, or epithelial cell of the body, if let's suppose it get infected, so then infected or disturbed, so then what happen, they will secrete some kinds of interleukin, as you can see. These interleukin are called as interleukin 4. This interleukin 4 will activate or migrate the helper T cell 2, or mainly they will first of all activate the helper T cell. So let's suppose this is the receptor, as you can see, and from there they will represent the portion of that helminthus. Helper T cell will come contact with this antigen. Let's suppose this is a helper T cell. Okay, so once they will contact with this antigen portion, and once the strong recognition done, then this is actually the naive helper T cell. Then this naive helper T cell will be converted into T helper cell 2, means this T cell will be matured. So this is helper T cell 2. Okay, this helper T cell 2, what actually this helper T cell 2 will decide? They will secrete interleukins. This helper T cell 2 will secrete interleukin, which we call that interleukin 4, interleukin 5, and interleukin 3. Okay, now this interleukin 4, what actually interleukin 4 will do? They will activate the B cell. Okay, once they activate and stimulate the B cell, then this B cell will be converted into plasma cell. Means again the same process will occur. This plasma cell will secrete antibody, and these antibody are actually IgE. Let's suppose they will secrete antibody which we called it IgE antibody against this parasitic worm. Okay, so they will secrete IgE antibody against this parasitic worm. Second, interleukin 5. What are the job of this interleukin 5? Interleukin 5 will activate the mast cell and eosinophil. Okay, mast and eosinophils. And all of you better know mast cell, eosinophil are the acidic cell for the other infection or for the pathogens, okay, or for the parasites, because they contain granules which contain acid like lysozymes, these things. So this interleukin 5 will activate these two cells. Then interleukin 3, they will activate the intestine to secrete mucus. Okay, so once let me write here, mucus means they will secrete mucus from intestine. Each mucus are actually kill the parasites. Now here one question arises in our mind, that parasites are very large and each cells and antibody are very small, then how it can kill the large parasites? Let's a little bit explain it. So if let's suppose this is the parasitic worm, as you can see, let me draw here. Let's suppose if this is the parasitic worm, so then what happened? As I talked that interleukin 4 is secreted, due to which IgE antibody will be secreted by the plasma cell. So plasma cell will secrete IgE antibody. IgE antibody will bind on the helminthus body. Phagocytic cell can't engulf this helminthus. So IgE antibody will bind it on their body, as you can see. A lot of IgE antibody are binded here. Once the binding is done, once this parasite are coated with this IgE antibody, after that what happen? Mast cell and eosinophil will bind here, because at the Fc portion the binding of the mast cell is perfectly, so mast cell will perfectly bind to this Fc portion, as you can see. Okay, so let's suppose these red are the eosinophils and these blue are the mast cell. After that what happen? The intestine will also secrete the mucus. Let's suppose these are the mucus. Now what happened? Let me a little bit explain that what happened. First of all, IgE antibody binded, a lot of IgE antibody bonded, and they will coat the parasites. Once it is coated by IgE antibody, once this parasite are coated with IgE antibody, after that mast cell eosinophil will bind to the Fc portion of each antibody. After that mucuses are continuously secreted, and second is these mast cell and eosinophil, their granules are burst, and once their granules are burst, means these eosinophil and mast cell will secrete their granules, and they will burst their granules, due to which the granules get internalized inside this parasitic worm. These granules means not granules inside the granules, the lysozymes will get internalized. There is not only a lysozyme, hydrogen peroxide are also present. Okay, so these hydrogen peroxide and lysozymes are actually acidic in nature. So each chemicals molecules, when get internalized, which are produced by eosinophil and mast cell granules, then they will ultimately kill this parasites. So that's how the parasitic infection will be under the control by the activation of each mast cell, eosinophil and antibody. So this is how the parasitic infection will be controlled. And it is done by this helper T cell 2. Now let's talk about the helper T cell 17. Now let's talk about helper T cell 17. This helper T cell 17 activated at that time when there is the extracellular infection, like bacterial or viral extracellular infection are occur. So at that time helper T cell 17 will be activated. So let me write infection. So how, let's suppose again in this case, let me draw here the normal cell. If this normal cell get infected by the bacteria or viruses, so then they will secrete, this is, let's suppose, infected, they will secrete interleukins 6 and transfer growth factor beta. These two cytokines are secreted at that time when the cell are infected or if there is extracellular infection occur. Okay, now after that what happened, they will activate the naive cells, naive helper T cells. So let's suppose this cell will also representing the antigen portion on their antenna, as you can see. So let's suppose this is naive helper T cell. This naive helper T cell will be converted into, once this interleukin fall down on this naive helper T cell, so then it will get mature, and after the strong recognition, it will be converted into T helper cell 17. T helper cell 17. T helper cell, helper T cell 17 is actually, as the name indicated, helper T cell 17 means they will secrete interleukin. They will then secrete interleukin 17 and interleukin 22. Okay. After that, what happened? This interleukin 17 will activate neutrophil, or means they will recruit neutrophil at the infection site. Refill at infection site. Second, interleukin 22 will activate the epithelial cell to secrete antimicrobial, means they will activate the epithelial cells to secrete what? Antimicrobial peptides against each pathogen. So this is the job of helper T cell 17. Okay, sometimes this helper T cell will also activate it against fungi, fungal infection or bacterial infection, extracellular infection. So this is a little bit information about the helper T cell 17. Now let's talk about the follicular helper T cell. So let me explain here. Follicular T helper cell. Follicular T helper cell is also the type of helper T cell which will be activated against HIV infection, or we can say lupus, diabetes. Let me write, it will be activated at different kinds of infection like HIV, lupus, type 1 diabetes, mellitus, and rheumatoid arthritis, means against the autoimmune disease. So this follicular helper T cell activated at that time if there is HIV, lupus, rheumatoid arthritis, or type 1 diabetes mellitus. So then against each infection and diseases, the follicular helper T cell will get activated. If any one of infection or disease occur, then it will activate it. Not as you better know that not each disease will be present at the same time. Any one of disease will be present inside the body. What happened after that? This follicular helper T cell will then, let's suppose the body got infected, or let's suppose the cell got infected, let's suppose. So once the cell, let's suppose this is the cell, it got infected, after that they will secrete interleukin 21. This interleukin 21 will activate the B cell. After that, this B cell will be converted to plasma cell, and then they will secrete antibody. But normally what happens? This interleukin 21 will actually guide the B cell to secrete antibody. To secrete what kind of antibody? Or to secrete those antibody which needed the body of the human or the body of the organism. So if this is the parasitic infection, so then it will secrete IgE antibody. If this is bacterial infection, they will secrete IgM. If this is the viral infection, they will secrete IgA, IgM, whatever. So actually interleukin 21 guide the B cell to secrete those antibody which are needed by the human, or which are needed. Okay, so we can say that these follicular helper T cell are actually, it is also important for the vaccination. It means if there is any kind of infection occur, so then we will take the blood of each patient, and then we will remove the serum, and after that we will use it for the vaccination process. It is again another large and huge topic, but we can say that follicular helper T cell are also involved in the formation of a vaccine or vaccination process. Why? Because it actually secreted those antibody those antibody which are needed. So that's all about the helper T cell, different types of the helper T cell, function of the helper T cell, means cytokines which are secreted of the helper T cell, and the function of each cytokines in detail. I hope you get idea about the helper T cell. Inshallah, in the next video lecture, I'm going to talk about the T regulator cell in detail. If you like this video, so make sure to hit the like button. If you understand, best of luck. If you didn't understand, if you have any kind of question in your mind, you can write each question in the comment. Inshallah, I will try my best to give you answer as soon as possible. So please like this video, share this video, and also subscribe the channel, and stay tuned, because in the next video we are going to talk about T regulatory cell. Thank you so much for watching this video.